3-in-1 program with the title Plant Breeding Classical to Modern Approach for Better Crops. Let me introduce myself. My name is Azeri Gautama Arifin, and I will be your MC for today's class. Before we start the class, I will remind you that in the end of the class, there will be a link of attendance form. Please do not forget to fill the form. And for the students of plant breeding class, there will be another link with a quiz in it. Please also do not forget to fill the form. Thank you. Selamat pagi, yang saya hormati Profesor Yukio Ozaki, yang saya hormati Bapak Ibu dosen pemulihan tanaman serta mahasiswa Fakultas Pertanian Universitas Brawijaya. Perkenalkan, nama saya Azeri Gautama Arifin. Saya bertugas sebagai MC pada acara hari ini. Sebelum acara dimulai, saya ingin menginformasikan bahwa akan ada link kehadiran pada akhir acara. Dimohon untuk Bapak Ibu dosen serta mahasiswa mengisi form tersebut. Terima kasih. Ladies and gentlemen, now we are going to listen to the expert opinion on role of biotechnology for plant breeding delivered by Professor Yukio Ozaki. Ladies and gentlemen, please welcome Professor Yukio Ozaki.
Thank you. Thank you for the introduction of myself. Then today I will start today's lecture by using the PowerPoint. Do you see the PowerPoint file?
Yes.
Is it okay? Yeah.
Yes,
then,
we can see it well.
Yes, then I will start. Then, good morning, everyone. Do you hear me? It is my third online lecture. Thank you.
It's very clear.
Yes, very nice. Yeah. This is my third online lecture in plant breeding. Thank you for Professor Dr. Arifin for providing me such a nice and honorable lecture. Last time I made a presentation of the plant breeding focusing on genetic-based theory. Yes, sorry. Today I'll talk about the role of biotechnology for plant breeding. There are two contents in this lecture. The first is inducing genetic variability, and the second is mutant and molecular selection. Firstly, I will explain inducing genetic variability. The phenotype, namely the observed trait, is the product of the genotype and the environment. The phenotype may be altered by altering genotype, environment, or both. When individuals from a clonal propagation are grown in the field, the plants will exhibit differences in the expression of some trait because of non-uniform environment. The field is often heterogeneous with respect to plant growth factors, nutrients, moisture, light, and temperature. Plants that occur in more favorable parts of the field or are impacted to a lesser degree by an adverse environmental factor would perform better than disadvantaged plants. That is, even clones may perform differently under different environments, and inferior genotypes can outperform superior genotypes under uneven environmental conditions. If a breeder selects an inferior genotypes by mistake, the progress of breeding program will be slowed. As you know, environmental variation is not heritable. However, it can impact heritable variation. Plant breeders want to be able to select a plant on the basis of its genetic nature, not growth environment. To this end, evaluation of breeding materials are conducted in a uniform environment as much as possible. Furthermore, the selection environment is often similar to the one in which the crop is commercially produced. Variability that can be attributed to genes that encode specific trait and can be transmitted from one generation to the next is described as genetic variation. Because genes are expressed in an environment, the degree of expression of a heritable trait is impacted by its environment, some more so than others. This figure shows the environmental effect on gene expression. In a class A, the environmental influence is small, and the phenotypes are distinguishable in the F2 generation. In class B, in contrast, the environmental influence is strong, resulting in more blurring of the differences among phenotypes in the segregation population. Heritable variability is indispensable to plant breeding. As previously noted, breeders seek to change the phenotype permanently and heritably by changing the genotype that encodes it. Heritable variability is consistently expressed generation after generation. For example, a purple-flowered genotype will always produce purple flowers. However, a mutation can permanently alter an original expression. For example, a purple-flowered plant may be altered by mutation to become a white-flowered plant. Genetic variation can be detected at the molecular as well as the gross morphological levels. The availability of biotechnological tools, for instance, DNA markers, allows plant breeders to assess genetic diversity of their materials at the molecular level. Some genetic variation is manifested as visible variation in morphological trait. For example, plant height, color, size. While compositional or chemical trait, for example, protein content, sugar content, content of a plant part, sugar content of a plant part requires various tests or device for evaluating them. It is impossible to improve plants or develop new cultivars without genetic variability. Once the objectives have been determined, the breeders then assembles the germplasm to be used to initiate the breeding program. Sometimes new variability is created through crossing of selected parents, inducing mutations, or using biotechnological techniques. Whether used as such or recombined through crossing, the base population used to initiate a breeding program must of necessity include the gene of interest. That is, you cannot breed for disease resistance if the gene conferring resistance to the disease of interest does not occur in the base population. There are three ways in which genetic or heritable variability originate in nature: gene recombination, modification of chromosome number, and mutations. The significant fact to note is that rather than wait for them to occur naturally, plant breeders use a variety of techniques and methods to manipulate and make these three phenomena more and more targeted as they generated genetic variation for their breeding programs. With advances in science and technology, for example, gene transfer, somaclonal variation, new sources of genetic variability have become available to the plant breeder. Variability generated from these sources is, however, so far limited. Genetic recombination applies only to sexually reproducing species and represents the primary source of variability for plant breeders in those species. Genetic recombination occurs via the cellular processes of meiosis. This phenomenon is responsible for the creation of non-parental types in the progeny of a cross through the physiological exchange of parts of homologous chromosomes. The cytological evidence of this event is the characteristic crossing, namely X configuration or chiasma of the adjacent homologous chromosome strands, allowing genes that were transmitted together, non-independent assortment in the previous generation to become independent. Consequently, sexual reproduction brings about gene reshuffling and generation of new genetic combinations, recombinants. Unlike mutation that cause changes in genes themselves in order to generation variability, recombination generation variability by assembling new combination of genes from different parents. In doing this, some gene associations are broken. Consider a cross between two parents of constructing genotypes large A, large A, small b, and large A, large A, constructing genotypes large A, small b, and small a, large B. A cross between them will produce an F1 of genotype large A, large B, small b. In the F2 generation population, and according to Mendel's law, the gametes large A, large B, small a, large B, large A, small b, and small b will combine to generate variability, some of which will be old, like the parents, while others will be new, unlike the parents, as shown in this figure. The larger the number of pairs of allelic genes by which the parents differ, the greater the new variability that will be generated. Representing the number of different allelic pairs by n, the number of gametes produced is 2 to the n, and the number of genotypes produced in the F2 following random mating is 3 to the n, with 2 to the n phenotypes, assuming complete dominance. In these examples, two new homozygote genotypes, small a, small b, and large A, large B are obtained. It should be pointed out that recombination only includes genes that are already present in the parents. Consequently, if there is no genetic linkage, the new gene recombination can be predicted. Where linkage is present, knowledge of the distance between gene loci on the chromosomes is needed for estimating their frequencies. Additional variability for recombination may be observed where intra-allelic or inter-allelic interactions. occur. This phenomenon results in new traits which were not found in the parents. Another source of genetic variability is the phenomenon of gene transgression, which causes some individuals in a segregating population from a cross to express the trait of interest outside the boundaries of the parents. For example, taller than taller parents or shorter than the shorter parents. These new genotypes are called transgressive segregants. The discussion so far has assumed diploidy in the parents. However, in species of higher ploidy levels, for example, tetraploid, hexaploid, it is not difficult to see how additional genetic variability could result where a rake interaction occurs. One of the tools of plant breeding is hybridization between divergent parents, whereby breeders selectively mate plants to allow their genomes to be reshuffled into new combinations to generate variability in which selection can be practiced. Be carefully selecting the parents to be mated. The breeders has some control over the nature of genetic variability to be generated. Breeding methods that include repeated hybridization, for example, reciprocal selection and recurrent selection, offer more opportunities for recombination to occur. The speed and efficiency with which a breeder can identify by selecting among hybrids and their progeny desirable combinations is contingent upon the number of genes and the linkage relationship that are involved. Because linkage is likely to exist, the plant breeders is more likely to make rapid progress with recombination by selecting plant genotypes with high chiasma frequency. It follows then that the cultivar developed with the desired recombination would also have higher chiasma frequency than the parent used in the breeding program. New variability may arise naturally through modifications in chromosome number as a result of hybridization between unidentical genotypes or abnormalities in the nuclear division processes, that is spindle malfunction. Failure of the spindle mechanism during karyokinesis or even prior that can lead the errors in chromosome numbers transmitted to cells, such as polyploidy. In this case, individuals with multiples of the basis set of chromosomes for the species in their cells appear as shown in this figure. Sometimes, instead of variations involving complete set of chromosomes, plant may be produced with multiple of only certain chromosomes of deficiencies of offer as called aneuploidy. Sometimes plants are produced with half the number of chromosomes in the somatic cells as called haploids. Like genetic recombination, plant breeders are able to induce various kinds of chromosome modification to generate variability for breeding. In nature, there exist two types of variation in chromosome number. In one type, called euploidy, the individuals contain multiples of the complete set of chromosomes, that is, characteristics of the species. The basic number X. In another, called aneuploidy, individuals contain incomplete set of chromosomes that may be equivalent to the euploid number plus or minus one or more specific chromosomes, as shown in the upper table. The state of having multiples of the basic set in the somatic cell in excess of diploid number is called polyploidy, and the individuals with such cells, polyploids. Polyploids are euploids. When euploid comprise multiples of the genomes, for example, duplicates of the genome from the same species, they are called autoploids, and the condition autoploidy or autopolyploidy. In contrast, when a combination of genomes from different species are involved, the term alloploid or allopolyploid, and similarly alloploidy or allopolyploidy is used. Alternatively, the term amphidiploid or amphiploid, and similarly amphiploidy or amphidiploidy is also used to describe polyploid with different genomes. It should be pointed out that autoploidy and autopolyploidy are extreme forms. It should be pointed out that autoploidy and alloploidy are extreme forms of polyploidy. Intermediates occurs between them on a continuum of genomic relationships. Stebbins called the intermediate segmental alloploids. Polyploids are named such that the prefix to the standard suffix refers to the base chromosome set as shown in lower table. For example, triploid refers to a cell with 3 genomes, while hexaploid refers to a cell with 6 genomes, 6x. In terms of general morphology, an aneuploid would resemble the original parent, whereas an alloploid would tend to exhibit a phenotype that is intermediate between its parental species. Autoploidy increases cell size, especially in meristematic tissues. Autoploids usually have thicker, broader, and shorter leaves. Other plant organs may increase in size compared to their corresponding part in diploids, an effect called gigas features. The gigas luxuriance contribute more to moisture content of the plant part than to biomass. The plants tend to be determinate in growth. The growth rate of polyploids is less than that of diploids. This may be due to their lower oxygen content than their diploid counterparts, as found for tetraploids. Polyploids tend to flower later and over a longer period. In grasses, autoploidy tend to reduce branching or tillering. Polyploidy also affect the chemical composition of plant parts. For example, artificial polyploidy increased the synthesis of artemisinin produced by diploid Artemisia annua twofold to fivefold in induced tetraploids. Similarly, the vitamin C content of vegetables and fruits has been known to increase following chromosome doubling. The nicotine content of tetraploid tobacco is about 80% higher than in diploid species. Autoploids generally have fertility problems and have poor pollen production. In some cases, reduction in fertility as compared to their diploid counterpart may be as high as 80 to 95%. This reduction in fertility is attributed to genetic imbalance following chromosome doubling that leads to disharmonies in development. Some changes in ecological requirements, such as photoperiod and heat requirement, have been reported in some species following chromosome doubling. The strategies employed in the breeding of polyploid are determined primarily on their origin. Harlan and DeWitt conducted from an extensive review of the literature that nearly all polyploids arise by the path of undedused gametes. They pointed out that the most common factor leading to polyploidy is the fusion of 2N and N gametes to form a triploid, followed by either backcrossing or selfing to produce a tetraploid. Further, they observed that the occurrence of undedused gamete is variable and pervasive in the plant kingdom. The undedused 2N gamete arises by one of two mechanisms: first division restitution, FDR, or second division restitutions, SDR, during meiosis as shown in the figure. Each mechanism has a different genetic consequence. In the FDR, the 2N gamete result from parallel spindle formation after the normal first division of meiosis. The cleavage occurs across the plane of the parallel spindles, producing dyad and 2 times 2N pollen. The genetic consequence of the mechanisms is that most of the heterozygosity of the diploid hybrid is conserved in the 2N gamete. In the SDR mechanisms, the first meiotic division is followed by cytokinesis, but the second division is absent. This results in a dyad with 2 times 2N gamete. However, in terms of genetic consequence, SDR results in significantly reduced heterozygosity in the 2N gametes. Researchers such as Bingham have proposed the fusion of the two FDR in 2N gamete to harness the heterosis that results in breeding potatoes. This heterosis can be fixed. The elite line produced will then be clonally propagated. Seed propagated species cannot benefit from this strategy. Plant breeders were initially attracted to induce polyploidy primarily because of the gigas effect, which increased cell size but also reduced fertility. This pro and con of the gigas effect make the induction of autoploid more suited to crops whose economic part is vegetative. The primary techniques for inducing autoploids is the use of colchicine, an alkaloid from the autumn crocus, Colchicum nail. Another chemical, Originate, is also used for autoploid production. These chemical compounds work by disrupting the spindle mechanisms in mitosis, thereby preventing the migration of duplicate chromosomes to opposite poles at anaphase. They are applied to growing regions of plants. Young seedlings or their apical meristems may be soaked in an appropriate concentration of the chemical for an appropriate amount of time. Axillary or subaxillary buds may also be used. Verification of the success of the treatment may be conducted in several ways. The easiest being visual inspection. Such an inspection produces only tentative results. Early physical evidence of polyploidy includes thicker and broader leaves, larger flowers and fruits, distorted growth, and shortened internals. Putative polyploid needs to be authenticated by more reliable, albeit more time-consuming method, such as examination of the pollen and the chloroplast count, flow cytometry, or chromosome count. For chromosome count, root tips and anther are popular materials to use. It should be noted that induction of polyploidy is not uniform throughout the treated materials. The convention is to recognize three histogenetic layers, L1, L2, L3, which may be altered differently. Guard cell reflects the L1 layer, while the cortical layer and reproductive tissues, for example, anthers, reflect the L2 and L3 layers, respectively. When gametes have the somatic chromosome number 2n, they are called the unreduced gametes. This condition occurs widely among angiosperms, and some researchers believe this to be origin of polyploidy plant species. Whereas artificially induced allopolyploids have fixed heterozygosity, sexual polyploid lacks these characteristics, and hence recombination occurs between the alien parental genomes. Also, because of recombination, introgression can be achieved. Successful introgression breeding has been achieved in species such as Alstroemeria, Lilium, Medicago, Sorghum, and Primula. 2N gametes may be detected in plants through approaches such as pollen size examination, flow cytometry, and progeny analysis. Intergenomic recombination is critical for introgression. The 2N gamete introgression techniques has been used successfully in potato, brassica, musa, Allium cepa, Lilium longiflorum, and others. Haploid contain half of the chromosome number of somatic cells. Anthers contain immature microspores or pollen grains with the haploid chromosome number. If successfully cultured, the plantlet resulting will have a haploid genotype. Haploid plantlet may arise directly from embryo or indirectly via callus. To have maximum genetic variability in the plantlet, breeders usually use anthers from F1 or F2 plants. Usually, the haploid plant is not the goal of anticulture. Rather, the plantlets are diplodized to produce diploid plant by using colchicine for chromosome doubling. This strategy yields a highly inbred line that is homozygous at all loci after just one generation. Methods used for breeding self-pollinated species generally aim to maintain their characteristics, narrow genetic base through repeated selfing over several generations for homozygosity. The idea of using haploids to produce instant homozygous by artificial doubling has received attention. Haploids may be produced by one of several methods: anticulture to induce androgenesis, ovary culture to induce gynogenesis, embryo rescue from wild crosses. The potential of haploid breeding to hasten crop improvement is attractive. However, the method is yet to become a mainstream breeding approach. Haploids cannot be obtained in the high enough frequency that is needed for selection. Further, haploids will express recessive deleterious traits. The method may not be cost-effective overall. Nonetheless, haploids have been used in breeding of many crops, including asparagus, barley, citrus, corn, grape, cucumber, pepper, peanut, wheat, and potato. Haploid breeding applies to species with polysomic inheritance, for example, potato, is different from that applied to species like barley and rice. Polyhaploids obtained from polyploids have polysomic inheritance and may be homozygous or heterozygous. In an autotetraploid-like potato, dihaploids may be large A, large A small a, or small a, depending on the genotypes of the tetraploid, which could be heterozygous, large A, small a, large A, small a, and large A, small a. Consequently, doubling of haploids will not always result in homozygosity unless dihaploids are used again for the production of monoploids. Mutation is the ultimate source of biological variation. Mutations are important in biological evolution as source of heritable variation. They arise spontaneous in nature as a result of errors in cellular processes, such as DNA replication or duplication, and by chromosomal aberrations. The molecular basis of mutation may be described by mechanisms such as modification of the structure of DNA or a component base of DNA, substitution of one base for a different base, deletion or addition of one base in one DNA strand, deletion or addition in one or more base pairs. In both DNA strands and inversion of a sequence of nucleotide base pairs within the DNA molecules. Mutation may also be induced by plant breeders using agents such as irradiation and chemicals. Many useful mutations have been found in nature or induced by plant breeders. For example, dwarf nutritional quality genes. However, many mutations are deleterious to their carriers and are hence selected against in nature or by plant breeding. From the point of view of the breeder, mutations may be useful, deleterious, or neutral. Neutral mutations are neither advantageous nor disadvantageous to individuals in which they occur. They persist in the population in the heterozygous state as recessive alleles and become expressed only when in the homozygous state, following an event such as selfing. Without adequate variation, plant breeding is impossible. To start a breeding program, the breeders must find the appropriate genotypes from existing variation or create the variation if it is not found in nature. Mutagenesis is the process by which new alleles are created. It is important to discuss mutagenesis as both a technique and a breeding method. The newly created mutant may be used as parents in future breeding programs, in which case mutagenesis is a breeding technique as a source of variation. However, an induced mutant can be systematically processed through conventional breeding steps to be released as a cultivar, hence making it a breeding method. Mutations arise spontaneously in nature and are pivotal in nature evolution. Spontaneous mutations produce novel alleles for the evolutionary process. Natural mutations have the benefit of being processed through the evolutionary process, whereby visible mutant become recombined with existing forms and become adapted under the under the guidance of natural selection. Mutagenesis can be used to create new alleles that can be incorporated into existing cultivars through recombination following hybridization and under the guidance of artificial selection. Modern crop production systems are capable of providing supplemental care of enables a mutant that would not have survived natural selection to become productive. As previously discussed, the significant number of commercial cultivars originated from mutations breeding techniques. Furthermore, the rate of spontaneous mutation is low, 10 to the minus 6 per locus. Artificial mutagenesis aims at increasing mutation rate for desired traits. Mutation may originate in the gametic or somatic cells. Gametic mutations are heritable from one generation to the next and expressed in the entire plant. However, mutations in the somatic tissue will affect only that portion of the plant, resulting in a condition called chimera. A chimera consists of two genetically distinct tissues and may produce two distinct flowers on the same plant. However, the dual color is impossible to reproduce by either sexual or asexual propagation. Commercial use of chimera is not attractive because the vegetative propagules must of necessary comprise both kinds of tissues in order to reproduce the maternal features. It is also well known that cells at the G2 gap phase and M mitosis stages are more sensitive to radiation than those at the G1 and S synthesis stages. In terms of gene actions, a mutation may be a recessive mutation, changes of a dominant alleles to a recessive allele, large A to small a. Dominant mutation, changes of recessive alleles to a dominant allele, small a to large A. Open pollinated species may accumulate a large amount of recessive mutant allele without any adverse effect. However, upon selfing, the recessive alleles become homozygous and are expressed, leading to the phenomenon of inbreeding depression. Using recessive genes in breeding takes a longer time because it requires an additional steps of selfing in order to identify and select the desired recombinants. On the other hand, dominant mutation manifest in the current generation, needing to no additional regeneration to be observable. Agents of artificial mutations are called mutagens. They may be grouped into the two broad categories, physical mutagens and chemical mutagens. The specific agent vary in ease of use, safety issues, and effectiveness in inducing certain genetic alterations, suitable tissue, and cost, among other factors. The principal physical mutagens are ionizing radiations as shown in the table. X-rays were first to be used to induce mutations. Since then, various atomic particles have been generated using nuclear reactors. Gamma radiation from radioactive cobalt is widely used. It is very penetrating and dangerous. Neutrons are hazardous and less penetrating, but they are known to seriously damage chromosomes. They are best used for materials such as dry seed, whereas the gentler gamma radiation is suited. Also for irradiating whole plant and delicate materials such as pollen grains. The relative biological effectiveness of fast neutrons is higher than for gamma rays and X-rays. The breeders is interested in identifying and using treatment with high relative biological effectiveness for maximizing the number of mutants produced. Treatment with high RBE have high ionization density. Modifying the treatment environment can increase the effectiveness of the treatment. Chemical mutagens showing this table are generally milder in their effect on plant materials. They can be applied without complicated equipment or facilities as compared to physical mutagens. The ratio of mutational to undesirable modification is generally higher for chemical mutagens than for physical mutagens. However, practical success with chemical mutagens lags behind achievement with physical mutagens. Usually, the materials is soaked in a solution of the mutagens to induce mutations. Chemical mutagens are generally calciogenic and must be used with great caution. One of the most effective chemical mutagens group is that of alkylating agent. Another group is the base analogs. Example are 5-bromolactic and maleic hydrazide. Other chemical mutagens commercially used are ethyl sulfonate, EMS, and EDES. Plant breeders who undertake wide crosses often need to manipulate the chromosome number to have fertility. To do this, the most common chemical mutagens used in colchicine, as mentioned in the modification of chromosome number. Chemical mutagens produce more gene mutations and less chromosome damage. Species that reproduce vegetatively or by apomixis may also be improved through mutation breeding. Vegetatively propagated species tend to be highly heterozygous. Consequently, selfing is accompanied by inbreeding depression. Mutation breeding offers a method of crop improvement whereby the genetic structure is largely unperturbed. Physical mutagens are more frequently used in such species than chemical mutagens. The plant parts targeted for mutagen treatment are those that can produce a bud from which a plant can develop. These parts include modified parts. The exposure to the mutagens must occur as early in the development of the bud and target the meristematic cell. A mutation in the meristematic cells is critical to avoiding chimeras, which can delay selection because the breeders may not be able to identify the mutant without propagating all the materials additional times. Chimeras are desirable in the breeding of certain ornamental species, such as African violet. Induced mutations in clones are of necessary dominant mutations, unless the starting materials was heterozygous and hence could yield recessive mutants. Furthermore, clonally induced mutants are primarily chimerical and started as sectional mutants, later becoming periclinal. The growing point commonly has 2 layers, some have 3. The outer layer generating the epidermis as well as some leaf mesophyls, while the inner layer generates the remaining part of the parent, as shown in this figure. The innermost layer, L1, has 1 cell per layer, while L3 has multiple layers. Each layer of the tunica corpus gives rise to specific anatomical regions of the plant body. L1. This is the first or outer layer of the tunica. It contributes to the epidermis in all organs. In dicot, this layer is colorless, and hence an L1 mutation is invisible. In monocot, it gives rise to a strip along the leaf margin. L2 layer, this is the second layer of the tunica. It contributes to the outer and inner cortex of stem and root, as well as the mesophyll in the outer region of the perimeter of the leaf. It gives rise to mesophyll of the petals and sepals, as well as internal tissue of the stamens and pistil, hence the pollen and seed. A mutation in this layer is what gets transmitted to the seed. L3, this layer contributes to the corpus and not involved in any part of the flower, fruit, or seed. Mutations, being one-celled event, result in part of wall layers exhibiting chimeras. A periclinal chimera is one that involves the entire layer, whereas a periclinal chimera involves only part of one layer. Periclinal mutations are stable. The number of initial cells in the layer in the growing tips determine the sectorial patterns. Sectorial patterns are mostly unstable, eventually producing periclinal structures. Because of this behavior, the breeders need to grow several clonal generations successively and select for the desired mutant, as well as vegetative stability. Vegetative stability is attained when uniformly periclinal lines of subclones have been isolated. In vegetative system, the treated plant meristem is called the M1V1, the next vegetative plant being M1V2, and so on. Secondly, I will talk about mutant and molecular selection. There are 3 steps in mutant and molecular selection: establishment of molecular marker, mapping of genes, and marker-assisted selection. Genetic markers are simply landmarks on chromosomes that serve as reference point of the location of other genes of interest when a genetic map is constructed. Breeders are interested in knowing the association of markers to genes controlling trait they are trying to manipulate. The rationale of markers is that an easy to observe trait is tightly linked, located in close proximity on the chromosome to a more difficult to observe and desirable trait. Selection is the method used to advance the breeding population. Hence, in a breeding program, breeders select for the trait of interest by indirectly selecting for the marker. When a marker is observed or detected, it signals that the trait of interest is present. Markers do not affect the phenotypes of the trait by SE. Genetic markers can be detected at both the morphological levels and the molecular or cellular level, which is the basis for classification of markers into 2 genetic categories as morphological markers and molecular markers. Morphological markers are manifested as adult phenotypes, the products of the interaction of genes and the environment. On the other hand, molecular markers are detected at the subcellular levels and can be assayed before the adult stage in the life cycle of the organisms. Molecular markers are assayed by chemical procedure of necessity. Morphological markers, while still useful in plant breedings, are less so because they are limited in number and also influenced by the environment. Further, the phenotypes of some morphological markers depend upon the developmental stage of the plant at which they are assayed. Heritable variation is central to the success of plant breeding. One of the goals of breeders is to first understand the nature of genetic variation in the plant species of interest, so it can be properly utilized for plant improvement. Molecular markers are used for several purposes in plant breeding. Gaining a better understanding of breeding materials and breeding systems. The success of a breeding program depends to a large extent on the materials used to initiate it. Molecular markers can be used to characterize germplasms, develop linkage maps, and identify heterotic patterns. An understanding of the breeding materials will allow breeders to select the appropriate parent to use in crosses. Usually, breeders select genetically divergent parent for crossing. Molecular characterization will help to select parents that are complementary at the genetic level. Molecular markers can be especially useful in identifying markers that co-segregate with quantitative trait loci to facilitate the breeding of polygenic traits. Rapid integration of simply inherited trait. Integration of genes into another genetic background involves several rounds of tedious back crosses. When the source of desirable genes is a wild species, the issue of linkage drug becomes more important because the drugged genes are often undesirable, requiring additional backcross to accomplish breeding objectives. Using markers and QTL analysis, the genome regions of the wild genotype containing the genes encoding the desirable genes can be identified more precisely, thereby reducing the fragment that needs to be introgressed and consequently reducing linkage drugs. Early generation testing. Unlike phenotypic marker that manifested often in the adult stage, molecular markers can be assayed at the early stage in the development of the plant. Breeding for compositional traits, such as high lysine and high tryptophan genes in maize, can be advanced with early detection and selection of desirable segregants. Unconventional problem solving. The use of molecular markers can bring about novel ways of solving traditional problems or solving problems traditional breeding could not handle. When linkage drug is recessive and tightly linked, numerous rounds of backcrosses may never detect and remove it. Disease resistance is often a recessive trait. When the source of the genes is wild germplasm, linkage drug could be difficult to remove by traditional backcross procedures. The recessive linkage drug was removed by using DNA markers, flanking the integration of preselect for individuals that were recombinant in the vicinity of genes. The lifespan of new cultivars can be extended through the techniques of gene pyramiding. For breeding disease resistance cultivars, markers-assisted backcrossing can be used to achieve this rapidly, especially for genes with indistinguishable phenotype. Plant cultivar identification. Molecular markers are effective in cultivar identification for protecting proprietary right as well as authenticating plant cultivars. Molecular markers are classified in various ways, including on genetic basis and operational basis. On the basis of genetic characteristics, molecular markers may be grouped into two general categories. The first is single locus multi-array codominant markers. Examples are RFLP and microsatellite SSR. Microsatellites are capable of detecting higher levels of polymorphism than RFLPs. The second is multi-locus single-array dominant markers. Examples are AFLP, amplified fragment length polymorphism, and rapid random amplified polymorphic DNA. Genetic variation in the genomes may be assayed at the DNA level or the protein level. Further, a variety of mutations in the genome gives rise to the variations that cause polymorphism in the population. In this figure, this figure shows the difference between the result of electrophoresis involving a codominant marker and a dominant marker, whereas a codominant marker can discriminate between homozygous and heterozygous genotypes. A dominant marker cannot. These mutations cause variation in the DNA sequence or cause certain sequence to be repeated. In view of the foregoing, molecular markers may be classified into three classes according to the nature of genome variation or the method used for assaying variation. Variation in proteins, allozymes. Variation in DNA sequence polymorphism, for example, RFLP. Variation in DNA repeats, for example, microsatellite. Various methods are used to detect molecular markers. In this regard, four distinct conceptual classes of molecular markers may be identified: enzyme-based markers, hybridization-based markers, PCR-based markers, and DNA sequence-based markers. The primary purpose of a traditional geographic map is to facilitate navigation and access to features and places or elements of interest in a geographic area. In genetics, the genome represents such an area, the chromosomes being the key landmarks, while individual genes represent the places or elements of interest. To facilitate plant breeding, researchers benefit from knowing the genetic landscape of the genome as it pertains to the relative position of genes along a chromosome. Because most traits of interest to plant breeders, in fact, most biological traits are genetically complex. Mapping or analyzing these complex traits, called quantitative trait loci, helps in understanding their genetic architecture and thereby facilitate their manipulation by breeders and other researchers. The ultimate goals of gene mapping is to clone the mapped genes. There are two conceptual ways of generating a map of genes: genetic mapping and physical mapping. In genetic mapping, the classical genetic tools of pedigree analysis is used to determine the relationship between individuals in the progeny of a cross. The underlying principle, Mendelian law, is that genes and markers segregate via chromosome recombination during meiosis, and the patterns of segregation can be analyzed to reveal the strength of association between genes. Called linkage analysis, the frequency at which two genes loci become separated during chromosomal recombination by crossing over is used to calculate distance between the loci on the chromosome. The map produced by this process is called linkage map or genetic map, and the process linkage mapping or genetic mapping. There are three basic types of physical mapping: chromosomal or cytogenetic, radiation hybrid, and sequence maps. These types vary in degree of resolution. Of these three, sequence maps have the highest resolution, being able to show genetic markers as well as the sequence between them. Whereas linkage mapping attempts to assign gene positions through breeding experiments or pedigree analysis, which are not precise. Physical mapping by sequencing provides absolute position of mapped genes. It is achieved via various methods, including the use of STS, for example, EST and SSLP for physical mapping. Radiation hybrid mapping relies on artificially induced by radiation breaks to determine the distances between markers. It is recommended that genetic and physical maps do be introduced for higher accuracy. Adequate polymorphism is critical to success of linkage mapping. Polymorphism is identified by the use of markers. Consequently, the next steps in linkage mapping is to identify polymorphic markers. The choice of suitable markers for mapping depends on the species being mapped and availability of characterized markers. Once identified, these markers are screened across the entire mapping population. To accomplish this, DNA is extracted from each of the plant selected. The expected segregation in different mapping populations has been discussed previously. Information on each plant is coded properly for the ensuring linkage analysis. Manual linkage analysis is feasible if only a few markers are being studied. Modern linkage maps constitution is a computerized operation, popular mapping package, including Linkage 1, G-Mendel, Join Map, Map Maker, and Map Manager. The last two being available free of charge via the internet. These computer software packages use the coded information from the segregating population to determine recombination frequencies. The basic calculation is a ratio of linkage versus no linkage, expressed as the log of the ratio. A LOD value of score measures the likelihood of linkage between two markers, a score of more than 3 usually being the cutoff minimum for mapping. A lot of these indicate a 1,000 to 1 odds in favor of genetic linkage. The researcher may vary the stringency of mapping by, for example, lowering the ROD score to detect a greater level of linkage. In developing new cultivars, plant breeders practice selection in segregating population on the basis of phenotypes. Most biological traits are genetically complex and their expression highly influenced by the environment. Selection efficiency and effectiveness, and therefore breeding progresses, could be adversely impacted if the trait of interest in the breeding program has low heritability. Selecting on the basis of markers linked to trait of interest could greatly facilitate the breeding of quantitative traits, except that QTL mapping remains challenging to allow its widespread application at this time. Nonetheless, molecular markers are used to considerable extent in plant breeding. Marker-assisted selection is the application of molecular marker in plant breeding. Molecular breeding is the application of biotechnological tools in crop improvement. As proven method of plant breeding, molecular breeding is the best considered not as a single breeding approach, like bulk screening or backcross breeding, but rather as a collection of tools and effort for improving trait phenotypes by a direct manipulation of the genotypes at the DNA level. The tools include genomic analysis, functional genomics, proteomics, and metabolic profiling. One of the more specific molecular breeding approaches is the use of molecular markers, especially DNA markers, for crop improvement. This approach is called marker-assisted selection. One of the earliest documented applications of marker-assisted selection in plant breeding was the breeding for resistance to soybean cyst nematodes. Since then, there have been an explosion in application of this breeding approach. Large-scale marker-assisted selection programs have been undertaken by CIMIT in Mexico for wheat improvement. Similar applications for this crop have been undertaken in Australia. Similarly, MAS for cultivar development in corns and many other crops have been reported. For application in marker-assisted selection, the key requirement of molecular markers include high degree of polymorphism, high reliability, ease of use, and cost-effective. Molecular markers are applicable to plant breeding forms, from program initiation to discrimination among the individuals in a segregating population, and cultivar identification. The key areas in which marker-assisted selection is advantageous over conventional breeding include distinguishing between heterozygous and homozygous genotypes, early generation discrimination, convenient screening, and reduced space for screening, and reduced breeding time. Especially reducing breeding time, early generation testing and erase of screening during selection may hasten the duration of the breeding program. In some, marker-assisted selection is simpler to conduct than phenotypic screening can be carried out at the seedling stage and allows single plant to be selected with high reliability, making for higher efficiency and accelerated lines developed in plant breeding. In some cases, using DNA markers in breeding may be more cost-effective than screening for target trait directly. Some of the suggested approaches to achieving higher success with marker-assisted selection include developing cost-effective and efficient marker systems by optimizing the method entailed in marker-assisted selection, such as DNA extraction and genotyping of markers. Using markers to reduce linkage drugs through recombinant selection, and screening of multiple traits per unit of DNA for rapid identification of genes functions in the major cereal crops. and continued use of MAS marker selection for breeding program that involves high priority traits that are difficult to expensive to measure. This is all of my presentation today. Thank you.
Thank you, Professor Yukio Ozaki, for a very great presentations. We have learned about ploidy and ploidy to the mutants and molecular selections. Okay, ladies and gentlemen, now we move to the discussion sessions. For the participants, please raise your hand or write your questions in the chat box. Please also mention your name and the class. Okay, Rahmat Julio raise his hand, please.
Yeah, thank you for the opportunity. First of all, good morning to Mr. Ozaki.
Good morning.
And all of my friends here, my lecturer. I want to ask about, from the previous, you stated that the percentage of mutation in plants is so small. So it can be done using the artificial mutation agent or mutagens. So based from some mutagens that you have mentioned, what is the most effective and widely used mutagen for the plant breeding? So, thank you.
Thank you for the question. And the effectiveness of mutagen is depend on the plant and in case of, for example, the induction of flower color variation, the physical mutagens are commonly applied for inducing the mutant, but the other types of the mutant induction, the chemicals are used because the efficiency of the mutation induction is larger and milder, not larger, but milder is in these chemicals. Is it okay?
So the use of mutagen is best from the plants. Is that right?
Pardon me?
He said that the mutagen is depend on the plants.
Yes.
Right?
Yes.
Yeah, thank you for the explanation, but I have one more question. Is that okay, sir?
Yes, please.
Yeah, can you explain to us more about autoploid, sir?
What kind of point?
Autopolyploidy.
Autopolyploidy.
Yes, sir.
There are two types of. Please wait. Not here. Do you see the slide? Do you see the slide?
Yes, sir.
Yeah.
Yeah. This shows the classification of polyploidy. In diploid, the genomic constitution is large A, large A. And euploidy, there are three types of euploidy, mainly two types, large A, large A, large A, and large A, large A, small a, large A, large A, large B. This is a polyploid constituting the similar or same constitution of genomes. In this case, we call autoploidy. And this type, there are two or more constitution of the genomes. It is called alloploidy. Is it okay?
Okay, sir. Thank you for the explanation.
Yes. Then when the plants large A are used for chromosome doublings, the large A genomic constitution will be obtained. And this is an example of autoploidy. Yes.
Okay, thank you, Professor Yukio Ozaki, for the
Yes.
explanations.
Yep.
Is there any other questions? Please, the audience, to raise your hands or write your questions in the chat box.
There's now my questions, please.
Okay, Mr. Arifin, thank you for the opportunity. First thing first, I want to apologize because I cannot turn on my camera because my unstable connection. So good morning, Mr. Ozaki.
Morning.
I want to ask about the first slide that you already taught us about genetic recommendation. So you said that only includes genes that are already present in the parents, but sometimes result in new traits which are not found in their parents. So my question is, how can these new traits appearing on these genes? Is it because of the condition of the environment, or it happened because the process of the establishing the genes itself, sir? Thank you.
Thank you. Thank you for your question. I think this is a example of the explanation, and in case of the plant pigment synthesis, there are plant pigments are produced in vivo under the control of the more than one genes. Then if the large A, both large A and large B are gathered in one plant, the plant pigment can be synthesized. But only one large A or only large B exist in the plant, then no plant pigment can be synthesized. That is one simple example of the gene combination effect. Is it okay?
No, if I'm right, I think the question is about, Sensei, sorry. The question is about introgressions. This phenomenon, other gene, what the cause of, example, environment or whatever, can cause the alien gene can enter in the genome. This is I think this is a phenomena, introgressions. What the questions like that?
どれできるかな。ホワイトボードできるかな。 Do you see the whiteboard? Then
Not yet.
Yes, we see the
yes,
whiteboard.
yes.
Yes.
This is phenylalanine, one kind of amino acid. Sorry. どうしようかな。これいかんのかな。This is the first time to use. Sorry. イレースですね。これか。sorry. いかん。そうか。あれで言ったらいいんだ。いいかな。
Yeah.
Sorry. よいしょ。
I think you can type. Typing is.
Sorry. Yes.
Yeah. The question is about, I think, about integration, Sensei. Why integration can happen in the genome?
Yeah. Integration of yes. This is a very simple types of the integration of genes. Then phenylalanine is a precursor of the anthocyanins, and phenylalanine can be changed to intermediate product by PAL gene, and intermediate products can be changed to anthocyanins. So red plant pigment by under the controls of ANS. Then, if the plant PP and PP AA and AA, if the plant have genes large P, and small a, phenylalanine can be changed to intermediate product, but it cannot be changed to anthocyanins. And in case of small p, and large a, phenylalanine cannot be changed to intermediate product. But the large p, small p, or large p, and large a, or large a, small a, in this case, phenylalanine can be changed to anthocyanins via intermediate product. This is a very simple types of the integration of genes. Is it okay?
So,
if I misunderstanding, please inform me. Only one gene cannot produce the precursor to anthocyanin, but the two genes, two locus are governs the anthocyanin synthesis. I think that is one of the interaction of two genes.
So which gene can be introgressed in this case?
Sorry. no, no, not. I am misunderstanding the integration of the meaning of integration. 電子移入。あ。ちょっと待ってね。when the interspecies hybridization occurs in plant A and B, and genomic constitution becomes to large A, large A, large B, and if the plants are back close to the by A, then the genomic constitution will be large A dash. But but I think I'm misunderstanding.
I think she asked about why there is phenomena that sometimes we cross a certain change, but the result, another change suddenly enter in this protein. This is I think this point of questions. Example, in the theoretically there is no such gene, but suddenly there is another gene or lion genes that come to the progeny. What is this because of environment or something? If I'm right, if I'm right, the questions.
Yes. Oh. I'm sorry, but I cannot catch the point of the question. Yeah.
It's okay, sir. Although I'm still confused, but I'm still trying to understand it. So thank you for the explanation.
If possible, please send the question by email via Arifin Sensei.
Okay.
I'm very sorry.
Okay, thank you, Professor Ozaki, and the participant has been joined this session. Time up. Let me close the class. Okay, ladies and gentlemen, this lecture about to come to the very end. I hope you have found the lecture on this 3-in-1 program are informative and helpful. Thank you very much. Till we meet again in 16 November.
Yes.
Okay.
So next
you
week.
have a great day.
Yes.
See you. Yes,
Yes,
next week.
Thank you so much.
See you. Wassalamualaikum warahmatullahi wabarakatuh. Thank you very much.
Thank you very much.
Sensei, sumimasen. Saki, great nandeska. I was coming late because I have to check the student lesson, another class. So.
No, not yet. No, not at all. Yeah.
Yeah. So I have received your number account.
Yes.
So I have not. So please wait for a moment because today or tomorrow my treasurer will send you.
Thank you so much. Yes. If the information is insufficient, then please inform me.
Yes, actually, I need some talk with you about the about lesson plan that we have a lesson plan, but we need review from others. That's when if you don't mind, please help me to review this lesson plan.
Yep.
We have about the production seed production technology and about the plant breeding, lesson plan.
Yep.
So I will send you if you're okay, then I will send you by email.
Yes.
And please review. Yeah.
Yes.
Of course, this maybe your facets from your facet, maybe give a mutual advantage in our lab, in our lab.
Yep.
Yeah.
I see.
So anyway, everybody is okay, Sensei?
Yes.
So
Yep.
who is your assistant? I forget. Your assistant professor? No.
Mizunoe, assistant professor. Yes. Mizunoe. Yes.
I think I don't knowledge.
Yes, I think you do not know him.
What about associate professor of you?
There are no associate professor in this lab.
Oh, just you and assistant professor.
Yes.
Oh.
Because the restriction rule is applied in the faculty, then there are very small number of laboratories where the three professor and associate professor and assistant professor exist. Most of the laboratory have the two professors, one professor and one associate or assistant professors. That is a very popular.
So, what about Hiramatsu-san? Hiramatsu-san?
Hiramatsu Sensei is in the Kyushu University, but not in the laboratory of horticultural science, but the other institute.
Really?
Yes.
So he's becoming a professor? Or?
No. He's now as a associate professor.
Associate professor.
Yes.
So Okubo Sensei as well?
Yes. Okubo Sensei as well.
Do you know the news?
I sometimes send the email, and he's
really?
he send reply very quickly. As is professor.
Because I'm sorry, I have no the current email of the Okubo Sensei, so I cannot.
Yes.
Yeah. So I have just Kyushu University email of the Okubo Sensei. I think is omitted, I think, right?
Yes. If you want to know, then I will inform you the Okubo Sensei's email.
Okay. yeah, of course.
Yes.
I want to say hello to Okubo Sensei because I just have the home number of phone number of his house. So I think is to be changed. I think no. Yeah. Yeah. Anyway, many thanks of your kind.
Thank you so much.
Great.
Yes.
Yeah.
Yes.
I'm sorry. See you, Sensei.
See you next week. Yes.
Yeah.
Yes. Thank you so much.